I'm a cell, that's me, neutrophil. Who's this? Topic is new genes, so fresh, so clean.
Notch 2 NL, don't put that DNase in your mouth. Ensim, stop eating your brother with filaments. You're gonna hydrolyze him. You, actually keep on doing whatever it is you're doing. Oh, hey, the viewer. Hey guys, why don't you introduce yourselves to them? Ah, perfect little angels. These little guys are humanity's newest genes. If you understand how evolution works, I'm sure you're willing to accept that over the course of the billions of years life has been around, that there may have been a few instances of new genes being born. But have you ever wondered exactly how these genes came to be? I'll tell you all about how new genes are born, and when these precious babies wake up, I'm sure they'll all be chomping at the bit to tell you what they do. Let's begin. New genes are basically always the results of happy little accidents. Such is the nature of biology, which is, at the chemical level, just a bunch of weird shapes bumping into one another. Some of those accidents occur during DNA replication of the cells that will eventually give rise to a full organism. One of the most well-known ways a new gene can be born is through a replication error that gives rise to a gene duplication. Because the role of the gene is still being filled by the original copy, the extra copy is free to mutate. We've actually talked about this in my video on the prion protein. Doppel and prion both arose from an ancient gene duplication event of the proto-prion gene. And we've also talked about this in my video about one of the longest running experiments observing E. coli evolution here. But how does gene duplication actually occur? In the past, I've generally explained one method of duplication in polymerase slippage. Polymerase, the enzyme that copies genetic material during replication, can slip and realign in funny ways, copying or deleting chunks of DNA. I chose to use slippage as an example because, well, it was easy to explain and animate. In reality, polymerase slippage does not usually result in the duplication of a whole gene, as slippage has a limited copying capacity. There are many ways gene duplication can occur, and I want to cover just two of them. Retrotransposition is a weird thing that can happen anytime a cell is making protein, not just during DNA synthesis. The first step of protein production is the production of messenger RNA, or mRNA, as clearly outlined in the central dogma. That mRNA is then sent off to the ribosome, where it is then read to make protein. In eukaryotes like you and me, the production of mRNA is a multi-step process. Eukaryotic protein coding regions of the DNA have different parts, introns and exons. Exons are the regions of DNA that actually code for a protein. Introns exist as spacers between exons. When DNA is read to make mRNA, both introns and exons are read. However, before mRNA is made into protein, the introns are cut out during a process called splicing. You might ask, why go through the trouble of this cutting and pasting? Organizing protein coding regions of the DNA into introns and exons allows you to cram more information in a smaller space. Think about it. Now you have a process by which you can mix and match exons to stitch together multiple different proteins. Pretty genius if you ask me. But what happens if this mature mRNA that has had all of its introns cut out gets interrupted before it gets to the ribosome to make protein? This is retrotransposition. Instead of being made into protein, this mRNA has a freak encounter with an enzyme called reverse transcriptase, whose job it is to take mRNA, convert it back into DNA, and insert it back into the genome. If the name of that enzyme sounds familiar, that might be because you've heard of it in the context of human immunodeficiency virus, HIV, a virus famous for inserting its own genetic material into your genome. An example of this happening is with the CDC14B retro gene, which, as its name implies, came from retrotransposition of the CDC14B gene at some point when we were great apes. CDC14B makes a protein that associates with the microtubules and aids in the regulation of the cell cycle. After millions of years of evolution, CDC14B retro gained a new function, associating instead with the endoplasmic reticulum. What it's doing with the endoplasmic reticulum? I don't know, but I'm guessing we'll find out soon. This next method of gene birth is basically gene duplication on steroids. Non-disjunction occurs when chromosomes fail to separate at some point during cell division. If this occurs during division of the progenitors of sperm and egg, this can cause one germline cell to be up a chromosome and the other to be down a chromosome. giving rise to a class of conditions called aneuploidy. Non-disjunction is not the most common form of gene duplication, but it can be the most groundbreaking evolution-accelerating event given just the right conditions. In fact, you, watching this video now, are the result of at least one non-disjunction event that doubled your entire genome. At some point, a replication event 404'd so hard that copied DNA never separated into another cell, doubling all of the cell's genetic material. In most cases, this is super deadly for the cell. Cells are generally not ready to support the existence of double the amount of genetic material for very long. But if somehow an organism manages to survive such a catastrophic event, evolution is then supercharged, as a redundant copy of every single gene enables a near infinite genetic sandbox. I honestly couldn't figure out all the details of how this fortunate disaster happened, but I can say that there is pretty strong genomic evidence that there were two historical whole genome duplication events that happened in between when we were looking something like this to when we were looking something like this. While the details of exactly which genes came into being by whole genome duplication versus chromosomal rearrangements is still being worked out, I think it's pretty incredible to realize that you being here is kind of a miracle on top of a miracle. Once again, whole genome duplication is really very bad news, except for some reason if you're a plant. Plants have a very high tolerance for whole genome replication, so much so that the organism with the largest genome happens to be the humble fork fern. Weighing in at a whopping 160 billion base pairs, the fork fern genome absolutely mogs the human genome. This fern's genome isn't diploid, it isn't tetraploid, it's octoploid. Absolute madness. If you want to know how our ancient ancestors survived our genome duplication events and why we think plants have a much easier time surviving theirs, why not leave a comment and tell me that's what you want to see. And subscribe so when I do make that video, you'll actually see it. And membership because you get free emojis to use during my streams and exclusive non-science content. Okay, that's my plug. So far, all the methods of gene birth have been through duplicating an existing gene and having a copy evolve over time. Sometimes, duplication events can rearrange genes to create new ones. Gene fusion occurs when a replication error brings two genes together, forming a hybrid gene. And if gene fusion can occur, then gene fission can occur too, splitting an existing gene into two bits. Exon shuffling can occur because DNA can get tangled during replication. When the tangled DNA gets resolved through a process called recombination, exons, the regions of DNA that code for protein, can have their positions scrambled, resulting in a weird Frankensteining of protein parts. Another way genes can be reshuffled into new proteins is from the action of transposable elements. Transposable elements, or transposons, or jumping genes are segments of DNA that can just leave the chromosome and insert themselves back in pretty much wherever. And if that sounds dangerous because it could potentially break something, you'd be right. In this way, perhaps the smallest pathogens around really are transposons, something that I should have covered in my last video on pathogens smaller than viruses. So yes, I will own my rightful razzing. I forgot transposons could count as pathogens. Just like a man-child who refuses to grow up and leave the house, transposons are glitchy, parasitic little leeches, contributing very little to the organismal project because most of them are non-coding and occasionally break things. But on occasion, transposons have their moments of glory. While they are generally considered to be neutral to harmful selfish elements, through a process called transposable element domestication, you too can make an honest entity out of these parasites. The tension between host and parasite can be resolved the moment both of these parties have something to gain. Through sheer statistical luck, a series of mutations could convert a once useless transposable element into something useful. Then, selective pressures can fix the transposable element into place, codifying it as a real gene. One of the most famous examples of this happened to the Transib transposon, named after the Trans-Siberian Railway. Back when we were all blobby fish, a Transib transposon found itself fixed into the genome, and in the evolutionary line of jawed fish, that transposon mutated to become RAG1 and RAG2. Transib was a transposon that coded for its own transposase, an enzyme that mediated its own cut and paste ability. A few mutations later, and this DNA cutting and pasting enzyme became two of the most important enzymes in all of immunity. That's right, all vertebrates that evolved from the jawed fish owe the existence of the adaptive immune system, that is the wing of the immune system that contains the T cell and the antibody-producing B cell, all to a domesticated transposon. RAG1 and RAG2 mediate the process of VDJ recombination, the technique T and B cells use to rearrange their DNA to generate new receptors and new antibodies in an attempt to beat ever-evolving pathogens through sheer statistical might. I outlined VDJ recombination in my video about how the immune system distinguishes between healthy microbes of the gut versus pathogens. It's a pretty good video, and you should definitely check it out. All of the ways that genes can be born described so far have been through modifying genes already present within the cell. But this isn't necessarily always the case. Occasionally, new genes can have their start as little gifts from the outside world. Horizontal gene transfer refers to the process by which a cell is given genetic material through a neighbor, as opposed to vertical gene transfer, which is the process by which a cell inherits genes through their parents. While horizontal gene transfer is an everyday occurrence for microbes, it's far less common for horizontal gene transfer to result in a new gene for more complex organisms. And while it isn't as common, that doesn't mean it's impossible. The syncytin gene was born out of an ancient viral infection that targeted primates, and was fixed and domesticated to make a useful protein crucial for embryonic attachment. But since I've talked about syncytin quite a bit on this channel already, I wanted to highlight a route of horizontal gene transfer that you don't really think about too often, between host and endo symbiont. It's not always easy to remember that contained within your eukaryotic cells are actually two sources of DNA, chromosomal and mitochondrial. Given our long history together, it would make sense that in the process of domesticating the mitochondria that some of their genes would end up in our chromosome. A gazillion years ago, the proto-eukaryote, probably archaeal in nature, gobbled up a bacteria, but through a freak accident, digestion was not completed. The bacteria might have had something to offer the archaea, and vice versa. This mutually symbiotic relationship deepened over evolutionary time as the bacteria started to lose its independent identity, becoming one with the host, giving rise to eukarya. But how would that bacteria begin that process of becoming mitochondria? At some point, this endosymbiotic bacteria gifted its host copies of its most important genes. Then, once those genes were no longer required for the protomitochondria to upkeep, they lost them over time. One such example is the mitochondrial polymerase gene, which makes a protein that helps the mitochondria replicate its own genetic material. This polymerase gene used to be stored in the mitochondria when it was, its own independent organism. But now, since the host has this gene instead, the mitochondria is completely dependent on us for replication. Depending on your perspective, you could see this as the mitochondria gifting protoeukarya a gene, offloading a gene they didn't want, or protoeukarya stealing a gene. Of course, the strictly correct interpretation is a mitochondrial gene ended up inside the host, but that's an answer for buzzkills. The last category is the one that I am the most excited to talk to you about, de novo gene birth. All of the mechanisms of gene birth up until now have been remixing genes that already exist, but on occasion, genes can be born out of the nether, the bubbling sea of chaos known as non-coding DNA. This idea of genes being born from basically nonsense garbage slop is completely non-intuitive. In fact, famed Nobel Prize winner François Jacob. In fact, famed Nobel Prize winner François Jacob. In fact, famed Nobel Prize winner François Jacob once wrote in his 1977 essay Evolution and Tinkering that the probability that a functional protein would appear de novo by random association of amino acids is practically zero. And now I, famed micro-celebrity on the internet, get to dunk on this bozo in this segment. Before I get too much further though, I need to make sure we're all on the same page when it comes to the term non-coding DNA. For a long time, this term was confused with the concept of junk DNA. We know now that non-coding DNA does not mean non-functional DNA. While non-coding DNA contains sequences that were never, ever meant to be read to make protein, they still do a lot of important things like making functional RNA, like the RNAs that make up the ribosome, regulating mRNA production through regions called promoters and enhancers, protecting your DNA from age-related degradation in the form of telomeres, and ensuring that your chromosomes are able to correctly separate during cell division through centromeres. We've also already talked about a type of non-coding DNA earlier in this video, the intron. Remember, in eukaryotes, protein coding genes contain both introns and exons, but when mRNA is processed, the intronic sequences are spliced out. Introns enable protein sequences to be mixed and matched, enabling higher protein diversity while keeping DNA sequences efficient. Don't get me wrong, there are still large swaths of DNA that have no known function, and these intergenetic regions tend not to be very well conserved at all, meaning that they can show high variability between individuals, between species, etc., etc. But all of these non-coding regions all share one important aspect. They were never, ever meant to be read to make protein. So what if, through a series of improbable events, they were allowed to make protein? The exact details and order of events here are a little hazy for me, but I'll try my best to outline one potential way the process by which non-coding DNA is given coding potential. There are at least 3 elements a stretch of DNA needs to be a real protein coding gene, including the DNA must be sufficiently close to a DNA sequence called a promoter. The RNA that gets made from that promoter must be stable, and the RNA that gets made from that promoter must have a start codon in the right place to be able to fit appropriately into the ribosome to get the protein production party started. Remember, DNA is double-stranded, meaning promoters can initiate transcription on either strand. A lot of the time, only one of the 2 strands has a protein coding region that checks all of those boxes downstream of the promoter in the direction that RNA transcription happens. However, this doesn't mean that transcription cannot happen on that other strand, it's just that those transcripts are often very short and very short-lived. But over time, a lot of time, these short-lived sequences can mutate away early termination sites, lengthening the transcript. It doesn't matter how long the sequence is if it instantly gets destroyed by quality control though. Through, you guessed it, more mutations, a transcript could obtain a poly-A tail, a short, roughly 6 nucleotide long sequence of mostly adenosines, which leads to the protection and stabilization of the RNA. Now we roughly have what we'd call a long non-coding RNA. For the most part, these non-coding RNAs will not be read to make protein, unless, through mutations, they acquire a start codon. The ribosome needs the start codon, typically a sequence of A, U, and G, to mark the beginning of protein production. The AUG has to be placed in a Goldilocks zone where it is far enough into the RNA for the ribosome to firmly grasp onto it, but not so far into the RNA where the ribosome is more likely to just fall off before starting protein production. You might be comfortable with the idea that mutations over time give rise to genetic diversity, but you might have a hard time accepting that de novo gene birth actually happens because it requires so many specific types of mutations, all to give rise to a single new gene. And you'd be right. However, biology is a numbers game. A study showed that for every unique sequence of coding mRNA, there are about 3 unique sequences of long non-coding RNA in humans. While the cell makes a lot more coding mRNA, the unique sequences of non-coding RNA blows protein coding RNA out of the water. This represents a bubbling vat of unlimited potential, a vast sea of RNAs each waiting for their day in the sun. It might not be today, or tomorrow, or even 1,000 years from now, but one day, one of these RNAs is going to make it big and be made into protein. Now, let's be realistic. A region of the gene that has no business being read to make protein is, in the vast majority of cases, going to produce RNA slop, which will then be made into protein slop. To really illustrate this, I'm going to generate a random string of amino acids to make a random protein and throw it into AlphaFold to show you just how unlikely a random peptide can form something functional. Remember that in order for a protein to be functional, in most cases you need some level of structure, usually a combination of alpha helices and beta sheets. If your protein ends up looking like nonsense spaghetti, there's a good chance it won't actually do anything. And it looks like our spaghetti is done cooking, and quite a bit more structure than I was expecting. That's actually kind of neat. Considering there are short peptides that are functional, like the antimicrobial peptides neutrophils make, this might be a genuine starting point for evolution to iterate on. And of course, once this gene actually exists, we've already heard about the other tools nature can use to further functionalize this young gene. Given enough time and luck for selection to do its thing, even random peptide slop can form a functional gene. This is the case for yeast gene BSC4, a gene that makes a protein that is involved in DNA repair. And for a while, BSC4 was the poster child for a gene that arose de novo. And it wasn't until some buzzkills pointed out that as sequencing databases got better and better, a potential path to a genetic ancestor began to open up. Look, I'm not a genetics expert, so I don't have a dog in this race, but I will say that we've already encountered this problem before. In my giant virus video, part of what made giant viruses so incredible was the fact that the majority of their genes are orphans, a cute way of saying that their genes have no familial relationship to any other known genes. This could be the result of a completely divergent evolutionary path, or it could be an indication that much of microbial life on Earth has yet to be sequenced. BSC4 and other de novo genes may suffer from the same existential crisis. Are all de novo genes potentially just related to something we've never sequenced before? Maybe Francois was right. Maybe de novo gene birth is all a scam. Most sources I consulted seem to agree that de novo gene birth has happened, but the details on how, which genes, and to what frequency this happens are kind of murky. Sounds like a pretty interesting field of research to me. Oh, the children are up. Let me go see if they'd be willing to introduce themselves to you. Hey there guys, the viewers out there. Do you want to come with me and introduce yourselves? They've been really looking forward to meeting all of you. No? Why not? Because they're unk? Well, I tried. Here are some photos. This isn't an exhaustive list of new genes in Homo sapiens, but just a couple of my favorites. This one is Notch2NL, which arose from a duplication of the Notch2 gene. Initially we thought, poor kid, doomed forever to live in the shadow of their extremely famous parent. But as time went on, this kid's shown themselves to be incredibly brilliant. Yes, Notch2NL takes after their parent as a development coordinating gene and has specialized in coordinating the growth of the brain's prefrontal cortex. This one's NSIM, which arose de novo as an antisense counterpart to its twin, MIC-N. See what they did there with the names? MIC-N's another member of a famous family of genes that coordinates growth, and SIM evolved to help its sibling regulate neurodevelopment. However, these two rascals often get into a lot of trouble together, as NSIM's tendency to amplify MIC-N has been linked to the development of brain cancers, neuroblastomas. And finally, this one's otherwise known as... Sorry. It's just these guys are so young nobody's bothered to give them a handy nickname yet. This little guy's still a bit of a mystery, and is a very strong candidate for a gene that's been born de novo. Much like the other new human genes, this guy's also involved in the development of the brain. I'm noticing a pattern here. We know that this gene does make a real protein, but we have no idea what it does. It does appear to be upregulated in Alzheimer's. Whether this is a gene that is protective or destructive in this context has yet to be seen. It's hard to be a biologist and not wonder where, when, and how it all began, and it could include the formation of new genes. I've heard your feedback in the last video, and don't worry, stay tuned for the actual beginning of it all, abiogenesis. That one will take me a while, but I can't wait to see you in the next one. And while these genes look great swaddled in their cute blankets when they get a bit older, I bet they look right at home in a Find the Neutrophil hoodie. That's the video, bye! Thank you to all my members who continue to support this channel. You guys are rock stars.